All right, it's my pleasure to introduce our keynote speaker of the day, Dr. Nicola Mason. Dr. Mason is a professor in the Department of Pathobiology and holds the James Gilmore Endowed Chair Professorship at the University of Pennsylvania School of Veterinary Medicine. Nikki received her veterinary degree from the Royal Veterinary College in London, and then we were lucky enough that she crossed the pond to Philadelphia to come and do her internal medicine residency at Penn Vet, where I first met her at that time. She then, after finishing her residency and becoming a diplomat of the American College of Veterinary Internal Medicine, she went back and did her PhD in immunology. She then went and did a postdoctoral fellowship in the lab of Dr. Carl June within the Abramson Cancer Center at the University of Pennsylvania School of Medicine. Today, as a faculty member, she is an inspirational role model and plays a critical role in mentoring research trainees as well as our clinical trainees, our students, and our residents. In her lab, Dr. Mason's translational research group focuses on a comparative approach to accelerate clinical implementation of effective immunotherapies for canine and human patients and improves the lives of both species. Specifically, her research interests include adoptive immunotherapy using CAR T cells, as well as CAR iNKT cells for B-cell malignancies, as well as osteosarcoma, and also the development of fully canine antibodies from phage display for translational research. Dr. Mason leads the first NCI-based canine cancer immunotherapy consortium that was supported by the Biden Cancer Moonshot Initiative. And her research using adoptive cell therapies is funded through multiple federal and private foundation mechanisms. Her pioneering work in live recombinant listeria vaccine in dogs with osteosarcoma earned her the One Health Award for excellence in promoting the One Health initiative. And for those of you who haven't seen it yet, her research program and its impact on One Health was featured in with 60 Minutes in an interview with Anderson Cooper. And it's a really heartwarming feature with an interview with one of the teenagers with metastatic osteosarcoma that underwent the phase 2 clinical trial of the modified listeria vaccine that Nikki developed and went through clinical trials with dogs. But today, we're going to hear more about her cell-based therapies to modulate the immune system.
All right, thank you so much, Sue. That was a very generous introduction. Let's put this pointer up here now. There's a pointer here, hopefully you can see that. So thank you. Hopefully everybody can hear and hopefully see me. Quite small. But what I want to talk to you today about is some of the work that we've been doing with autologous as well as allogeneic T-cell therapies. And I'm going to focus on the use of those therapies for cancer. And then at the very end, I'm going to sort of bring it back to talk very briefly about some of the other ways we could use these genetically engineered cells. And really that's what we're going to be focusing on, is how we can genetically engineer T cells, as well as iNKT cells, to what we call harness the power of the immune system and direct it towards cancer to produce deep, durable remissions and possibly cures. So these are my disclosures. I'm not going to be talking about anything listed here today. But when we actually talk about harnessing the power of the immune system, we hear that that again and again. And I thought to myself when I was putting this together, how could I actually sort of visualize the power of the immune system? How powerful really is it? And as I was sort of thinking about this, I was reminded of some of the work, or some of the patients that I had seen in my internal medicine residency with rheumatoid arthritis. And so you can just look at these radiographs, and I don't think you need to be a clinician to recognize the amount of destruction that is present here in these particular patients with rheumatoid arthritis and autoimmune disease, where the immune system is redirected against self-antigens and engages, this is actually a CD4 T-cell mediated disease, it engages the rest of the immune system to produce cytokines, to produce autoantibodies, and to completely destroy bone and cartilage. So if you're in doubt as to how powerful the immune system can be, I think this is a really nice visualization of that sort of power. And you can think about if we could redirect that towards neoplastic cells, we would have a very powerful system to perhaps induce these durable, long-lasting cures. So let's sort of take a step back and think about this cancer immunotherapy revolution. And as you can sort of see here on the left-hand side, this is actually the front cover of Science magazine back in 2013, when cancer immunotherapy was voted the scientific breakthrough of the year. And there were two reasons for that. One was the recognition of the role of checkpoint inhibitors that I'm not going to talk too much about today. And the second was the identification that we could actually genetically redirect the specificity of T cells in something we call CAR T cells, which I'll talk about, to very specifically target tumor cells and eliminate them. But really, when we think about cancer immunotherapy, it's been going on for not just decades, for hundreds of years, in fact, thousands of years. People have been thinking, perhaps without knowing back then, of using the immune system to destroy tumors. And you can see from this immunity cycle the way we sort of think about this. We start off with agents that can destroy the cancer. So this could be radiation, it can be chemotherapy. What we're trying to do is release tumor antigens that then get picked up by antigen-presenting cells. Those antigen-presenting cells will present tumor antigen, activate the T cells, which will go off around the rest of the body to find the tumor cells and eliminate them, again then further releasing more tumor-associated antigens to propagate that cycle. And so we'll see an amplification of this cycle, a broadening of the T cell response in this way. But the truth of the matter is we've really been sort of focused on the left-hand side here of this cycle. We've been thinking about giving patients tumor antigens in some sort of immunogenic lysate with some sort of adjuvant to try and sort of start this cycle off. And other people have developed antigen-presenting cells that are preloaded with antigen and give those to the patient. And really, they have not been very effective. And so people sort of sat down and said, well, what do we really want to do? We really want to give the patient T cells. That's where we're trying to get to. So why don't we give patients T cells that are specific for the tumor antigen? And this is really where this concept of CAR T cells came around. And these are just two photographs here of Jim Allison and Tasuku Honjo, who won the Nobel Prize a couple of years back now for their work in checkpoint inhibitors. And I'm going to be bold and stand up and say that I think that probably a Nobel Prize is coming for the second breakthrough here, which is CAR T cell therapies. Although I have to say it's not going to be me that receives it, just so you know. So what are these CARs? What are these chimeric antigen receptor T cells? Well, basically, these are T cells that have been engineered to express a chimeric antigen receptor. And that consists of an antibody fragment, actually the variable heavy and variable light chains of a monoclonal antibody that are joined by a flexible linker that then has a hinge domain, a transmembrane domain, and co-stimulatory and T-cell stimulatory signals. So we have this CAR construct. And that CAR construct can be transduced into these T cells and essentially redirect the specificity of those T cells against the antigen that monoclonal antibody is specific for. And somewhat remarkably, when these CAR T cells come into contact with a cancer cell or any cell expressing that antigen, they will become activated. They receive the first and second signal through that CAR. They start to produce cytokines and proliferate and kill. So So when you actually give a dose of CAR T cells, you actually have the only drug that I can think of that actually increases in dose after you've given it, because these cells will proliferate and proliferate, so you have more of them. And this is the way that these cells are utilized. A patient will come in, will undergo apheresis, the T cells will be selected, they'll be activated, the CAR will be put into these T cells using lentiviral or retroviral technology. They'll be expanded ex vivo and then adoptively transferred back into the patient in an autologous system, and then those patients will be monitored. So it sounds a little bit crazy, sounds a little bit space-agey, and the question is, does it work? And the answer is, when we look at B-cell malignancies, the answer is yes. So when we look at these Kaplan-Meier survival curves, and these are patients with B-cell acute lymphoblastic leukemia or patients with B-cell non-Hodgkin's lymphoma, you can see that the survival of these patients is really dramatic. And now I'm going to tell you that all the patients on these clinical trials are relapsed refractory patients. So they have nothing left to try. So they've gone through the chemotherapies, they are out of options, and then they receive a single dose of CAR T cells directed against CD19, which is a B-cell antigen. And these CAR T cells will go in and eliminate all the B cells in these patients, healthy B cells as well as malignant B cells, but you can live without your B cells. And so you have 6 months survival here of 90% in acute lymphoblastic leukemia. It really is dramatic. And when you look at B-cell non-Hodgkin's lymphoma, we see a similar sort of effects, but the response rate is lower. So we have sort of half of these patients that don't respond, and then we have half of these patients that do respond. And you'll notice that if you do respond, you seem to have a complete and durable remission. And I think we all agree, if we could see our canine patients, 50% of our dog patients, having complete durable remissions after a single dose of a drug, in this case CAR T cells, that would be remarkable. So that sort of led us to think, well, could we actually do this in dogs? And also, if we were to do this in dogs, would we see the same sort of response here that may then enable us to look at next generation therapies to address those patients, perhaps in dogs as well as humans, that don't respond to CD19 CAR T cell therapy? So we set about generating a canine CAR T cell production sort of facility, as it were, and a workflow to make canine CAR T cells. And this was quite an undertaking, to say the least. But basically, we followed what was happening in the human field. So we would take canine patients. We would either do a large blood draw to get the T cells, or more commonly, these patients would undergo apheresis. And you can see a patient undergoing apheresis here. This is the machine that we use. We would get the product, and actually you can see here, this is sort of the collection of these cells. It's like a giant expanded capillary tube, and you can see on the top all the white blood cells accumulating, and then periodically they'll be pumped up into this bag here, and then we can do our T cell negative selection, activate our T cells, use a retrovirus to put in that CAR, expand those cells up, then wash them, and this is the little pellet here, and then adoptively transfer them into our dogs after they have received a preconditioning regime of cyclophosphamide. So the question then is, can we actually do this? I'm sorry if this hasn't come out particularly well, it looks blurry here, but basically what you can see here using flow cytometry is that we get a 60% transduction rate in this particular dog, and of those CAR T cells, 60% of them are CD8 positive T cells. We've got some CD4s in there as well. So what this tells us is we have canine T cells that are expressing this CAR, and this is a CAR that is specific for CD20, which is a marker on B cells. And when we take those cells and co-culture them with targets that express CD20, we can see production of interferon gamma. So here against a CLBL1 cell line, we see production of interferon gamma, and against another canine cell line that we've engineered to express CD20, again, these cells get activated, and they get activated when we co-culture them with B cells. And this is an antigen-specific response that's going through the CAR because we don't see activation when we don't have the target antigen present. And we can cryopreserve these cells, and then take them out of thaw and stimulate them again, and they produce interferon gamma in an antigen-specific way again. We can also proliferate them in response to targets in the green line here and the red line that express CD20. They will proliferate, but they don't proliferate well when we don't have the CD20 antigen. And then they kill very effectively here. So we're looking at B cells that express CD20, a B cell line. We get very efficient killing, almost 100% killing with an effector to target ratio of 4:1, whereas if the target is not present, we don't really see that killing. So what this is telling us is we can make CD20 specific T cells using this redirection, this engineering redirection, and those cells are in fact functional. So now we can put them into dogs, and we can ask that question. If we actually can genetically redirect these cells and put them into canine patients with B-cell non-Hodgkin's lymphoma, do we see therapeutic advantage? Do we see therapeutic effect? And the idea here is that the schema is depicted here. We were taking either treatment-naive dogs, or where the owners didn't want to treat with chemotherapy, or those dogs that had relapsed with CD20-positive diffuse large B-cell lymphoma. They would undergo apheresis. We would make their CAR T-cell product here. The patient would undergo lymphodepletion with cyclophosphamide, and then we would infuse a CAR T-cell product, an autologous product in there, and evaluate what happened to these patients. We've done about 13 of these dogs now, and I'm just going to show you a couple to sort of demonstrate some of the things that we're seeing. So this was a boxer, a 4-year-old male castrated boxer called Harley, and he had stage 5A B-cell non-Hodgkin's lymphoma, so he was heavily leukemic. And we can talk about that at the end, but I think that was quite important. It was very difficult to make a CAR T-cell product from this dog because all his lymphocytes were B cells. So it was challenging, and we were only able to make a product that would give him 1 × 10⁵ CAR T-cells per kilogram. We're aiming for a log or 2 logs above that. So this dog got a very, very small number of CAR T-cells. But we took serial aspirates of his lymph node on the bottom here and of his peripheral blood, and then did that at the days shown along the top. And of course, this dog, after his preconditioning with cyclophosphamide, a single dose of cyclophosphamide, didn't receive any more chemotherapy. So you can sort of start to see what happens, and what happens in the peripheral blood is reflected in the lymph node as well. That as you go through time, this dog's off chemotherapy now, so you can sort of see the B cells are sort of coming up, and then we start to see this switch. And now we start to see the number of CD20 positive on the x-axis, B cells, CD79A on the y-axis, start to decrease. And by day 138, this dog has no more CD20 positive cells in his body, not in his peripheral blood and not in his lymph node. We've actually managed to eliminate this dog's CD20 positive cells. And this tells us that this approach can work. But what happens? Well, we see that now we still have B cells present in this dog, but they no longer express CD20, and this dog's disease progressed on clinically, but he was progressing with a CD20 negative B-cell lymphoma, which was extremely disappointing, to say the least. So what was happening there? So we took an aspirate or some peripheral blood from this dog. We have a CD20 negative population here, and then we have some cells that still express CD20. And we performed a PCR to amplify up CD20, and you can sort of see it's not very clear, but in row 3 here, when we look at lane 3, when we look at the CD20 negative population, we see a down regulation of this expression, and we see an additional amplicon here at 552 base pairs. And when we sequence that, what we can see is following treatment, the CD20 of this dog changes. And we've lost exon 4 and we've lost exon 5, and that encodes for the extracellular loop of CD20 that our CAR T cells target. So this dog has undergone some sort of splice event where the target of the CAR T cells has dropped out, and this is exactly what happens in children treated with CD19 CAR T cells, and one of the key reasons in which these patients will relapse with antigen-negative disease, and we see it happening in the dog. Interestingly, I haven't got got time to show you this, but interestingly, this clone actually was present at the beginning of treatment. So what we did is we actually eliminated the CD20 positives, and then the CD20 negatives came through. And that means we have to start thinking about a bispecific approach for these patients. Do we see cytokine release? That's something that's a problem in humans. Yes, the answer is yes, we do. And this is another patient treated with 5 million CAR T cells per kilogram, so more what we're trying to get to. Happy little corgi running around, 72 hours later, not so happy. And what's happening here? Well, when we looked in his blood, looked clinically, this chap has a temperature just shy in the blue here, just shy of 106 degrees. And when we looked in his peripheral blood, we could see evidence of that lentiviral vector that's incorporated into his cells, and we see that's the peak activity. So his CAR T cells are expanding, his temperature is going up, and that is then consistent in cytokine beta array, looking at these serum cytokines, with production of IL-6, MCP-1, a bit of IL-2 as well, and some of these other cytokines, a profile that is very similar to what we see in humans with CRS. Now, people are spending a lot of time and money trying to make models of CRS for CAR T cells, and I would argue that if we can get this right, and if we can get sustained proliferation of these CAR T cells, the dogs are essentially a model for canines, for humans, and we can start to think about how we can modulate this response. But the truth of the matter is that in all of these patients, we haven't really seen good, solid clinical responses. So these patients, as you can see here in his lymph nodes, the lymph node size drops down, and then after that chemotherapy, comes back up again. So we haven't really seen the clinical effect we want to date. So why is that? Well, we may have inefficient preconditioning. We may have a failure of these CAR T cells to appropriately expand and continue to function. We're going into a pretty hostile tumor microenvironment, very solid. Lymphoma is a hematological malignancy, but we have big solid lymph nodes that have a very immunosuppressive tumor microenvironment. And we've seen those antigens get downregulated or even spliced out, and we recognize T cell dysfunction as well. And on the left here, you can see the reasons for why CAR T cell, CAR-19, fails in human patients with B-cell lymphoma. And you can see this relapse here with antigen-positive disease. That's usually associated with a loss of the CAR T cells. Relapse with antigen-negative disease, I've shown you that in the dogs. But 30% of these patients have a primary resistance. And one of the thoughts that why that occurs is because of checkpoints that are present in these solid tumors or hematological malignancies. And one of those checkpoints is PD-1, PD-L1. So this is a homeostatic mechanism. Activated T cells will express PD-1, and they interact with PD-L1, and that acts to turn off those T cells and sort of drives those T cells to exhaustion. And we think that that's actually quite important in why these CAR T cells may not be effective in solid malignancies and in perhaps these lymphoma patients. And so what's the evidence that PD-1, PD-L1 axis is present in B-cell lymphoma? And I'm showing that here, and this is actually Steve Dow's work, 2 papers that he's produced, showing PD-L1 expression on malignant B cells within lymphomas. So you can see a lot of these cells express PD-L1. We also actually see some PD-L1 here on the T cells that are present in those lymph nodes. And when we look at T cells present in those malignant lymph nodes, we see they are expressing PD-1. So we have our T cells expressing PD-1, we have our tumor cells expressing PD-L1, and that's going to interact and turn off these T cells. And we also know that interferon gamma upregulates PD-L1 on tumor cells. So you can think about your T cells going in, starting to kill these tumor cells, producing interferon gamma, and the only thing that happens is PD-L1 comes up and turns off your T cells. So we wanted to explore whether that happened in CAR T cells, and is this a reason why our CAR T cells are less effective than we would like them to be? And so in order to do this, we had to generate some target cells. We did this with a scaffold cell, a human erythroleukemic cell line, K562 cells. They're just a sort of scaffold cell that's very easy to transduce. We got them to express canine CD20, and then we got a next line to express CD20 with PD-L1, so we could start to ask questions about what does PD-L1 do on those targets. And we took our CAR T cells, CD20 CAR T cells, co-cultured them with these target cells at different effector to target ratios, and we're able to show that we get upregulation of PD-1 on these T cells when they see their target, CD20. It doesn't really matter at this point whether there's PD-L1 on the target or not. These CAR T cells get activated through their CAR and they upregulate PD-1. So what happens then when they see PD-L1 targets? And this is what we're seeing here. So we're looking at CD4 CAR T cells and CD8 CAR T cells against targets that don't express PD-L1 and targets that do. And this is in 4 healthy dogs. And you can see that there's a decrease in the proliferation of both CD4 and CD8 cells. You can see when we look at cytotoxicity, this is against CD20 expressing cells, showing here with our 4 healthy dogs. And then here, we're looking at cytotoxicity against the PD-L1 cells. So again, it's telling us that PD-L1 expression on targets is going to shut these cells down. They don't proliferate as well, they don't kill as well, and in fact, they also don't produce as much interferon gamma and IL-2. So PD-L1, PD-1 interaction is not good. And we know that that's the case in humans, and people are actually now trying to use CAR T cells with drugs like pembrolizumab, which is an anti-PD-1, to block that interaction to try and make those CAR T cells more effective. But could we sort of be smarter again and start to think about engineering these cells so that when they actually receive a PD-L1 signal, rather than turning the T cells off, we're going to turn the T cells on even more, and we're going to sort of enhance their function. And we can do this with a genetic switch. So what we've designed is a PD-1 on the surface here that's actually linked to a co-stimulatory domain in the T cell. And we've put that into the T cells along with the CAR. And so now what we would hope that happens is rather than that PD-1 engagement turning the T cell off, when the PD-L1 hits the PD-1, we're actually going to turn that cell on, and we should have enhanced T cell function. And so that's the concept here. So I'm just showing you here flow cytometric plots showing that we can get CAR expression in CD4s and CD8s. That's our control. And then you can see we've got our CAR here with our switch. And we obviously have nice expression of the CAR and nice expression of the switch as determined by PD-1 expression. And then we also have a mutant switch that doesn't signal. So PD-1 is there, but the CD28 switch doesn't work. And so you can see we get comparable expression of CAR with the wild-type switch and the mutant switch. We have a little bit less expression here of the CAR without the switch receptors, and that's been shown in humans as well, and it's not quite clear. I've got some ideas of why that is. But we certainly see CAR expression, and we see expression of the switch. So the question is, can we enhance the activity of these cells with our genetic switch? And the answer is yes, we can. You can see here the CAR with the wild-type switch being put against PD-L1 targets. That should turn them off. But now, because we've got the switch in there, we actually see more interferon gamma, more TNF-alpha, and more IL-2 in these dogs. We don't see that effect with the mutant switch. So this is dependent on active signaling through that switch receptor. We also can see enhanced proliferation in these triangles here. This is our wild-type switch against PD-L1 targets in both CD4 and CD8 T cells. So again, we're seeing an enhancement now where we should see suppression. And in cytotoxicity, it's a little sort of different here. We certainly see an inhibition of cytotoxicity with our CAR alone. And we don't see augmentation of cytotoxicity, but we do see a sort of an elimination of that negative effect. And we need to sort of look at this a little bit more carefully. So that's all well and good, and we're excited now to take that and those CAR T cells with the switch receptor into the dogs. We haven't done that yet. We're hoping to get that started soon, so we can see if we can actually get better clinical responses. But the truth of the matter is you can engineer these cells all you like. What's really important is the fitness of the T cells. So if you're starting off with a T cell that isn't particularly fit, you're probably not going to do very well. And this has actually been shown in the human clinics with CAR T cells, that if you actually look, this is work from a colleague at the medical school, looking at human patients with CLL treated with CAR-19 cells, and he looked at those those patients that underwent complete remission or partial remission with CAR T cells and found that those CAR T cell products were very much enriched for memory properties and had low levels of exhaustion. It sort of makes sense. You've got a much healthier product. Whereas if your cells were more sort of effector cells and highly exhausted, then your CAR T cells probably weren't going to be that great. And he could actually take that back one stage further and just look at the apheresis products before you've even generated the CAR T cells in those patients, and would actually be able to demonstrate that when you look at your peripheral blood, if you have a high level of central memory T cells, stem central memory T cells, CD8 positive T cells, you're going to do better. So that's great. So we can say, well, right, we're going to select those patients that have really healthy T cells, and hopefully they're going to go on and do really well. And we know that there are a number of different factors that affect T cell fitness, like age, disease burden, previous chemotherapy. So that's great if you've got sort of a healthy population of stem central memory in your peripheral blood. But if you don't, what are you going to do? That's going to be a serious problem for you. And this is really where I think the holy grail of adoptive cellular therapies lies, in us being able to produce allogeneic CAR T cells, off-the-shelf types of therapies that you can just go to, like we do for red blood cells, and say, oh, I need this particular pack of red blood cells. Now you can go and say, oh, my dog has lymphoma, I'd like a CD19 CAR product, or my dog has glioblastoma, I'd like an IL-13 receptor alpha-2 product, and you can get them from these large tanks, and in they go. So the advantages are fairly obvious, right? With an off-the-shelf, on-demand therapeutic, we can start to think about what is the optimal donor, what do we really need there, and we can get the best product, and everybody can get that product. There's immediate availability, and that's a problem, obviously, if you don't have immediate availability when we make autologous products. Sometimes human patients, and certainly the canine patients waiting a few weeks, may not have that time. We have product standardization. Everybody gets the same product. It's really hard right now to know why one patient responds and one patient doesn't, because everybody's getting a different drug. Everybody's getting their own autologous T cells. So that would be nice. Cost and time savings, that's obviously important. And you can actually start to use different combinations of cells. But we can't do this with T cells, because if we took allo T cells and put them into a recipient, we're going to get life-threatening graft versus host disease. We're going to get rapid elimination of those T cells, and those are major barriers. And so now people are sort of starting to do CRISPR editing, taking out the T cell receptor, taking out class 1 and class 2, putting the CAR in, and then using these cells. And we've done some of that using human CAR T cells in dogs, but I don't have time to show you that. But one of the issues here is that these are heavily genetically edited cells, and we don't really know about the safety, we don't really know about the efficacy of those cells. So is there another way that we could employ to actually have these off-the-shelf, on-demand therapies that we would really like to be able to utilize without the need for genetic engineering? And so this is an area that we're very interested in now, in exploring this idea of these invariant natural killer T cells. And so this is a sort of an alternative off-the-shelf platform that we're looking into. So what are these cells? Well, they're a super rare subset of T cells. You can see here they're 0.01% of your T cells are invariant natural killer T cells. And what the name suggests is that they have an invariant TCR alpha chain, and they recognize glycolipid that is presented in the context of an MHC class 1-like molecule called CD1D. And that is monomorphic. So my CD1D is the same as all your CD1Ds, and my invariant TCR chain is the same as yours. And what that means is I can take iNKT cells and put them into a recipient without worrying about graft versus host disease. So now we don't have to worry about editing out that T cell receptor. These cells are a sort of combination of natural killer cells, very rapid response against microbes, for example. They're able to produce cytotoxic granules like granzyme B and perforin. They kill through Fas ligand as well. They have a broad range of activities and cytokines that they produce, and they also like to home to tissues, which is exactly where we want them to be if they're going to go and kill the tumor cells. And they're sort of, an interesting subset of cells. So as I said, they have this invariant T cell receptor that recognizes glycolipid expressed by CD1D, and CD1D is expressed by many cancer cells. It's also expressed by tumor-associated macrophages. So we could actually think about these T cells going in, these iNK T cells going in, and actually starting to eliminate the tumor-associated macrophages and modulate the tumor microenvironment. But they also produce cytokines like Th2 cytokines. They produce IL-4. They interact with CD8 positive dendritic cells, and I'll show you this actually in vivo. And that will produce TGF-beta and can induce regulatory T cells. So the thought process is these cells can actually induce tolerance to themselves. So we have this hypothesis that we could take these invariant natural killer cells and actually adoptively transfer them into MHC mismatched individuals without the requirement for genetic engineering, and actually get them to persist. And is that true? And we wanted to ask that question in the dog. So the first question was, do we actually see iNK T cells in the dog? And the answer is yes. So these iNK T cells are recognized through this germline recombination of a TRAV10 and TRAG18. This recombination event makes up this CDR3 region of the TCR alpha chain. This is the sequence that we see in the humans, and this is the sequence in the dogs. And so we hypothesized then that tetramers, so CD1D molecules loaded up with this glycolipid with a fluorophore on the end of them, could be used to identify canine iNK T cells. And the answer is yes. And I have to say that the work I'm going to show you with these iNK T cells is done by a very talented postdoc, now research assistant professor in my lab, Dr. Antonia Rotolo. And I have to say she came to me and showed me these plots and said, aha, dogs have iNK T cells. And I was like, oh, sure, we're going to be able to work with that. There's not so many cells there. But nevertheless, she said, yeah, I'm pretty sure that we can make this work. And so she took these cells and she co-cultured them with feeder cells expressing CD1D and this alpha-gal with a little bit of IL-2. And you can see in the space of just 9 days, we were able to expand out these iNK T cells, and they really do have an incredible proliferative capacity. So we actually did this from 4 donor dogs. We made 4 individual iNK T products at clinical scale. And we cryopreserved those cells until we had all 4. And then we thawed them, expanded them again, and started to look at their capabilities, their cytotoxic responses. So our 4 donor dogs, these are relatively young, 2 to 6 years of age, male and female, donor dogs, healthy dogs. And you can see that we can start to proliferate them here now. These are undergoing sort of 8,000-fold change. They've already undergone about a 1,000,000-fold expansion in the original production of these infusion products. But what you can start to see is that these donors vary, right? 2 of them can proliferate a lot, 2 of them don't proliferate very much. And when we look at cytotoxicity, we find that donor 3 here kills very nicely, but donor 4 has sort of not such a great cytotoxic activity. So what we're starting to explore now is differences. We're starting to see differences in these products. So we're starting to think, what then is the best way, what is the best optimal donor for us in terms of iNK T cells? So in order to sort of drill down on this, we did single-cell RNA-seq on these 4 infusion products. And we were particularly interested in looking at central memory, because central memory has been shown, as I showed you with Joe Fraietta's work, to actually correlate with deep and durable responses and persistence of autologous CAR T products. So we were able to identify 3 clusters in this single-cell RNA-seq that were enriched for central memory characteristics. And so what we did is we looked at the frequency of iNK T cells from each donor that expressed these memory characteristics in each of these clusters. And you can see very much so that when we look at donor 3 again, we can see that the iNK T cells in these clusters, there was a lot of central memory characteristics. And we can see that again looking at sort of the average expression level of central memory features. We also see again in donor 3 more effector memory, less exhaustion, and more telomerase activity. So based on this, we thought to ourselves, well, donor 3 is the donor that we want to go with. Can we safely transfer 400 million of these iNKT cells, there's no CAR here at the moment, can we safely transfer those into MHC mismatched donor dogs, recipient dogs? And so we set up this now. Only 4 dogs in this study, and only 2 of those dogs got iNKT cells, so we're looking at very small numbers, and these are all healthy research dogs. So we took a bone marrow biopsy, and we took a couple of bone marrow biopsies and aspirates from these dogs, because we know these iNKT cells go to tissues, so we know we may not be able to find them in the peripheral blood. And these dogs received low-dose Cytoxan, 250 mg per meter squared, just a little bit of lymphodepletion. And then 2 of the dogs received iNKT cells. One just iNKT cells alone, and the other iNKT cells with alpha-gal, which is a glycolipid agonist that causes proliferation of iNKT cells, and also TBI, very low dose total body irradiation at 2 gray. And that was because it's been shown to induce tolerogenic dendritic cells and induce some proliferation of iNKT cells. So we wondered if that combination would help the persistence of these unedited cells. And so, and then we also had controls. We had 1 dog that just got Cytoxan and alpha-gal alone, and 1 dog that got Cytoxan and TBI. And you can sort of see now what happens in the peripheral blood. So with TBI alone, we see all of these cell types drop. You see that in the red here. With alpha-gal, remember that's what causes proliferation of iNKT cells, and we see it in the blue, not much effect on lymphocytes, but we see these iNKT cells starting to come up. And we certainly see Tregs coming up. So giving this alpha-gal agonist, we can start to see the effect on Tregs in vivo, and we hope that that will allow these cells to persist. And then when we give the iNKT cells, we sort of see something rather similar in both of these. We see some activation of lymphocytes, as shown here in this peripheral blood slide here. We see iNKT cells going up. We give 400 million iNKT cells, so in both of these dogs we can see that happening, and they seem to sort of persist out for about 29, 30 days. But of course, this could be the recipient's own iNKT cells. We don't know that these are the others. NK cells seem to come up a little bit, and again, we can sort of start to see these Tregs sort of starting to increase here. What's most important, there was no graft versus host disease, there was no cytokine release. And so could we see persistence of these cells? And we took advantage here of this concept that the donor was female and the recipients were male. So we could use Exist, which we heard about yesterday, a long non-coding RNA that's expressed by the active X to actually track those female cells. And you can see here, using qPCR, we can actually see these cells in the peripheral blood, female cells, so these we know are the donor cells, out till about 29, 30 days. And in the bone marrow, we can see them out to about 4 to 5 weeks. So that was exciting to see. Finally, we were able then to say, well, let's have a look in the peripheral blood. Let's see if we can expand iNKTs to increase the sensitivity. And then in those expanded iNKTs, let's see if we can see female cells. So that's exactly what we did. We took blood at 2 different time points with one of our iNKT recipients and another one here with the TBI and alpha-gal. We expanded them out, and then we looked within those expanded profiles at the donor cells. And you can see now we're starting to see these cells coming out at 78 days, at 118 days. So we're looking at months now. We're in the sort of 3 to 4 month range. And just to confirm that they indeed were the donor cells, we did sequencing of MHC class II alleles, and we're able to identify those donor-specific alleles. So we have these cells persisting now without the need for heavy lymphodepletion, without the need for exogenous cytokines, and without the need for engineering. So just in summary, I think, and now I don't have time to tell you about the CAR T cells, but we've been able to put CARs in these iNKT cells, and we're sort of gearing up for a study in metastatic osteosarcoma. So I think harnessing the power of the immune system is possible. It's possible in humans, it's possible in canine T cells. We can start thinking about these strategies to engineer these cells to overcome some of the immunosuppressive factors within the microenvironment. Obviously, developing these safe and effective off-the-shelf, on-demand therapies therapies is going to be very valuable for both humans and canines. And we're starting now to sort of dissect out what is the best molecular signature, perhaps, that would give us the best iNKT product. We don't know about safety and persistence of these multiplex edited cells, so that's why we're sort of moving a little bit away from those and trying to leverage the natural biology of this rather unique subset of T cells, which hopefully I've shown to you we think we can use in a fairly safe way. And then just to finally mention that engineered T cells is not just cancer. If you're not interested in cancer, that's fine. These are now being utilized, CD19 CAR T cells, remarkable results in patients with refractory lupus, just getting an injection of these CD19, eliminating their B cells and early plasma cells, and undergoing clinical remissions, infectious disease, fibrosis, cardiac fibrosis, idiopathic pulmonary fibrosis, as well as senescence. And so with that, I'm not sure, I hope I'm on time, but with that, I'd just like to acknowledge all the people, particularly Antonio Rotolo, who's done the iNKT work, and Sho Yoshimoto, who did the PD-1 work, and many other members in the lab across the Penn community, and funding here through NIH, and we just recently got a UO1, which I need to add on there. So thank you very much for your attention.
Incredible work. Thank you very much. Do we have any questions? Yes.
Thank you, Dr. Mason. That was a great talk and really interesting data. I was just curious, so CD19 CARs have been observed to be having neurological toxicity and B-cell aplasia along with a CRS. Those were the two concerning side effects. I was curious if your CD20 CARs had similar adverse effects, and if not, what else did you observe in terms of adverse effect?
Yeah, so we have not seen any evidence of ICANS, which is the neurotoxicity that's associated with CD19 CARs with our CD20 CAR. But I will say that I don't think that these CARs are effective as they should be yet. So we're not seeing the sustained expansion that we see in human patients, particularly with leukemia. And I think when we do see, and we've got a few reasons why we think that's the case, but I think that when we do see that, that's really when we're going to need to be looking. And I sort of, I'm hypothesizing that we're going to see similar sorts of things. You know, we see this CRS, but it's very mild. You know, it's not the same type of CRS that we see in the human patients that are often in ICU for 3 or 4 weeks in a coma from their CRS. But again, I think that when we start to see these cells taking off, I think we're probably going to see CRS. Whether we'll see ICANS, we'll have to see. We've also developed a CD19 CAR now, so I don't know if it recognizes a similar epitope. I don't know, but we've got a CAR that's redirected against CD19, so we'll be looking at that as well.
Okay, yeah, it was really exciting. I cannot imagine even this how huge work it is. I would like to ask several questions, but yeah, just a comment. Yeah, actually, you because we sometimes say that we develop CAR Ts against like tumor-specific antigens, but yes, it's important to say that these are not actually tumor-specific antigens. Yes, that you of course you said it, but it's you get rid of all cells. Yeah, so of course it is associated with huge risk, of course, also, and not so easy to choose antigens to attack. With B cells it's okay, yes, because they at least in humans, but with the other options it's not so clear. And I wonder because you came up with this really nice idea with PD-1. Combining CAR against CD20, with this, like blocking PD-1, but actually, what you showed from the first dog, you get rid of CD20 cells. So actually, it was not that CAR T was not effective; it was effective, yes. So it is, yeah, like.
Yeah, no, that's a good question. So, and we can talk more about it afterwards, but yes. So I think that shows that you can get rid of those CD20 positive cells. That dog was heavily leukemic, and the other dogs that we've treated have not been leukemic. And I think that there's I think that that's relevant, because I think when these CAR T cells go in, I think in leukemic patients, the first thing they see is B cells, and they start to get activated and they start to expand. And I think that that's really very important for these cells to be able to engraft and have persistent activity. So that dog lost all his B cells out at day like 150 plus. So I think that is important. But I think when they're in an environment of this tumor microenvironment, I think that's much, much harder for them to function.
All right, thank you very much. I'm sure that there are many more questions because it was a fascinating lecture, but you can find Dr. Mason later. Thank you very much.